molecules. Every other cell type makes TNF from scratch, just like other cytokines are made from scratch that takes 24 to 48 hours to be made. But we have been interested for many years on the effect of flavonoids on mast cells, and we had published a number of papers, I'm indicating 2 here, where we had shown that select flavonoids, especially quercetin, can actually block the mast cells. Flavonoids are antioxidant, which is good. They're anti-inflammatory, which is good. A new paper published about 6 months ago in PNAS showed that they're actually neuroprotective. They mimic the action of BDNF, brain-derived factor. And we have shown that they're mast cell blockers. So in this particular case, we used one particular flavonoid called luteolin, one of the richest sources of which is olive oil. And you can see actually that in response to substance P, you get a lot of mitochondrial fragmentation and movement to the membrane. And if you actually pre-treat for 5 minutes with luteolin, increasing concentration 50 and 100 micromolar, you can block this. And you can block it 100% if you go to basically a little higher than this. And you can block every cytokine that is being released from the mast cells as well. Now, why we were doing these studies with another little grant we got from NIH, recently we started actually trying to see if luteolin administration to animals could alter their ability for post-exercise fatigue. And I'm not going to have time to show you these results, but I can tell you that we can increase actually the ability of the animals to swim actually about 5 times by just giving them an IP injection of about this much luteolin. While we were doing this paper came out that absolutely fascinated me. They had an animal model just like the one we used, just like the one that Ben told us to use, and they actually showed that not only they can undo the symptoms of chronic fatigue, but they can induce some biochemical markers and oxidative markers in the brain, in this case by giving olive extract. Now, the reason why, not only I come from Greece, so I love oil, but it so happens that flavonoids are very lipophilic. So we actually put them on olive kernel oil. That's the oil that you get after you crush you get actually the oil, you're left with pits and flesh. The olive kernel oil is actually a little thicker, much richer in polyphenols. So we make rudimentary liposomes, and that's what we gave to the animals. So I actually jumped the gun, if you wish, and I convinced a little company down in Florida to put together luteolin along with 2 other flavonoids for reasons that we don't have time to go into, and they made actually a dietary supplement that is available under the name Neuroprotect. So I will finish by saying, number 1, I'd love to have CSF and blood to measure actually free-floating mitochondrial DNA in such patients, and I'd love anybody who would like to do a little pilot study using Neuroprotect. There are 2 PAs out from NIH for exploratory trials actually that would allow us to use something like this, except that I don't see patients and I depend on you. Thank you.
Okay, I think we have some time for some questions for Theo.
So you said prevented in your cell cultures that this prevents the degranulation?
It blocks degranulation. It actually blocks de novo synthesis of cytokines as well.
Huh,
so
cool.
It blocks IL-8, IL-6, TNF, histamine, etc.
So we have some data from our exercise study that shows TNF being released post-exercise. So that would have to come from the mast cells, you're saying?
Well, I mean, the mast cell might be the first cell to release it. Other cells make TNF. But for instance, in a number of other studies, some of which we published, if you stress an animal for 30 minutes in a plexiglass immobilizer, which we used to do in previous studies, you get very high TNF actually in the blood. And if you do the same in mast cell deficient animals, you don't get any. So at least in animals, TNF, mast cells seem to be a major source of TNF. But other cells make TNF, as you know.
So how might we prove that?
Well, there are ways of doing it. First of all, you can block, mast cells. Unfortunately, there aren't that many.
These are humans.
Yeah, humans.
Okay.
There aren't that good inhibitors of mast cells. There's only one, cromolyn sodium, that is only about 10% as active as luteolin, for instance. So one could try to block the mast cells, but have in mind that no one knows that luteolin is only specific for mast cells, so you must still be blocking other cell types as well. So the answer to your question is it might be impossible to show that it is only the mast cells in the humans. The only thing that talks towards your question is that, as you know, there is this group of patients called mastocytosis. They have a lot of mast cells. Interestingly, they don't have allergies, but they have tons of other problems that we've been discussing today. And about 80% of them or so, in a number of publications, complain of chronic fatigue. They have not been identified as chronic fatigue syndrome, but they complain of chronic fatigue. So if that's an indication, you have a live experiment. You have these patients that have tons of mast cells, and most of them seem to have chronic fatigue.
But in the patients we do have, say with this exercise model, if we looked for the mitochondrial byproducts or degradation post-exercise and correlated it with TNF expression,
that would be great. That would be absolutely great. And that's the kind of study that I was thinking about, to do it exactly that, do the exercise.
So we have those specimens, we'd be happy to collaborate.
Absolutely, yes. And this might not be a systemic phenomenon. It might be more localized to the brain. These are isolated cells that I showed you, and then animal experiments that I alluded to. So it could well be that it is the mast cells in the diencephalon that are acting up, in which case CSF might be the best place to pick it up rather than the serum.
James.
Yep, we did. So
Jim's got your stuff.
So what effect does it have on the mitochondria in the muscle?
okay, that I don't know, but I think it might be a double jeopardy. Assuming that everything we heard yesterday is correct, and I don't have any reason to doubt it, let's say that the mitochondria are fine. There's no major mitochondrial problem. And let's assume that we have 100 mitochondria. If 50 of the mitochondria are actually diverted from producing energy, you have part of the jeopardy. The second part would be that we'll be dumping mitochondrial DNA, which is immunogenic, so you get actually both without necessarily having a functional problem. Now, on top of it, of course, we could have some susceptibility, some genetic problem that we'll be hearing that would make things even worse. Or we might have a combination of triggers. It might be the exercise, but it might be that they've been exposed to something environmental or other, because as I told you, the mast cells respond to all kind of hydrocarbons. They respond to mercury, they respond to aluminum. So I'm not trying to say that they're beginning at the end, but many of us feel that the mast cell might be the first response of the body to some stressor, physiological, psychological, or whatever. For instance, a wonderful paper was just published in Immunology that they show interleukin-9 is released immediately upon either apoptosis, necrosis, or damage to any cell type. They tried about 10 different cell types, and they showed that the only cell that was activated was a mast cell. So I think that there will be much more that we'll be finding, except that it's almost impossible to study it in the brain, where maybe the action is.
So you're saying there would probably be a reduced efficiency in the muscle plus inflammation?
Correct, absolutely correct.
And one further question, the calcium, what effect is it going to have on total body calcium?
That I don't know. We know that calcium is absolutely mandatory for activating the mast cells. Originally we thought that the mitochondria might actually translocate to the cell surface to buffer or allow calcium to stay there to allow secretion. But unfortunately, the mast cell in culture responds within 15 seconds. And the confocal microscopy we have access to cannot do any faster than that to see if, in fact, there are local regulation of calcium ions. But I would imagine that if let's assume that the mitochondria are buffering calcium and that they're actually being dumped outside, then there might be fewer of them to actually buffer calcium. But that's a wild stretch.
Theo?
Yes.
So what would you expect to find in spinal fluid in patients with chronic fatigue
If
syndrome?
in fact the action is in the brain, and if it is the brain mast cells that are activated, I would expect to find higher levels of free-floating mitochondrial DNA in the cerebrospinal fluid, and potentially TNF, and we've already heard of, at least in 2 cases, TNF was high in CSF, even though it was flat in the blood. But I would love to, for me or anybody else, to do it at the same time. In other words, have both serum and CSF from the same patient run at the same time.
Theo?
Yes.
Just informational, what's the half-life? How long-lived are these cells?
I'm sorry.
Half-life of the mast cell.
The mast cell is actually extremely long-lived, but it's not circulating. It's found in tissues.
Okay.
An experiment was done by Jacques Powder at Albert Einstein long time ago, where he actually injected colloidal gold in peritoneal cavity of rats and killed them every month until they died. The idea was that they take colloidal gold, and if they die, then there shouldn't be any colloidal gold left. And colloidal gold was actually inside the mast cells until they died of old age. So under at least sterile, normal circumstances, the mast cells seem to be staying around forever. The other thing that we absolutely know is we used to think that they're like, one-shot cell. They secrete and then they die. Under normal conditions, both in the body as well as in tissue culture, within 24 hours, it will restock the granules and be ready to fire again.
Theo, some time ago we showed that NPY also activates mast cells, but releases PGD2,
yes,
newly formed mediators, but not preformed mediators. I don't know if you can comment on that and whether you've tried that, because PGD2 has a lot of CNS effects. And
you're absolutely right. Not only you, Levi Schaefer from Jerusalem and a few others have shown the same thing. The only reason why we didn't look at those is because the proper way to measure them is a little complicated, and I've never done that. And what is available kit-wise is not as reliable because these things get metabolized, as you know, very quickly. But I wouldn't be surprised at all if molecules such as PGD2 or some leukotriene may also be involved. We already know that leukotrienes, at least leukotriene C4, activates the HP axis. We already know histamine activates the HP axis, and IL-1 and TNF activate the HP axis. And I'm saying this because a number of these patients, as we know, worsen by stress. Stress that might not necessarily be related to cortisol, but may be related to CR8 that is released actually from the median eminence.
Okay, thank you, Theo. Those were
Thank you.
very intriguing